All right, Ninja Nerds, we're going to continue our discussion with the inflammatory response. This is going to be part 3. If you haven't seen part 1 and you haven't seen part 2, click up here in the right corner for our immune system playlist. We'll have all the parts available for that. All right, so let's go ahead and continue. So lastly, we were talking about this phagocytosis process, right? Because we've already went over the entire basically inflammatory response with all of the vascular and cellular events, and then we went through this phagocytosis mechanism. Now, phagocytosis is not the only way that we can kill these bacteria or these microorganisms. There's another way. We're still on the innate immune system, so non-specific. Now, we're going to talk in this discussion here about what's called complement proteins. So you know the liver actually is constantly synthesizing specific proteins called complement proteins and pushing it into the circulatory system. And these guys are normally just circulating in our actual plasma, but then whenever there's a site of inflammation, just like our white blood cells can squeeze out, so can these molecules. So these complement proteins, when activated, they can also squeeze out here into the tissue spaces and go to fight this microorganism. So let's go ahead and go right into those. So there's 3 pathways of the actual complement system, and we're going to go over each one individually. So here's our bacteria. And look at this guy, he's mean, right? All right, so he's kind of mean here. And what he's gonna do is, he's gonna present those antigens. Remember, he has those antigens up on his surface? So here's that antigen right there. Here it might have an antigen right there. Look, he looks like a devil. All right, you know there's another thing that we're gonna talk about, we'll talk about it in future videos. There's a lot of antibodies that are constantly circulating within our plasma or within our tissue fluid. Some of those antibodies might leak out here, and they might be specifically memory antigens. And what do I mean by that? There might be there might be antibodies that have been previously exposed to this bacteria before. All right? And let's say that it could be IgM antibodies or IgG antibodies specifically. If they've already been infected by it, it would be IgG antibodies. And we'll talk about that when we talk about antibodies. But this IgG antibodies will bind on to this actual bacterial antigens. When they bind onto the bacterial antigens, there's a specific portion here on the antibody, which is called the Fc portion. The Fc portion is very, very attractive for our complement proteins. So now here's what we're going to get into. We're about to get into some hardcore stuff here. Ready? So first one, these proteins are circulating in the area, right? They just so happen to run in to this antibody right here, the Fc portion of it. This complement protein right here that first binds is called C1. That's the first complement protein. Then after that, another complement protein binds here. This complement protein is called C4. Then after that, guess what else we have? We have C2. Then it goes C3. But here's where it gets funky. There's an enzyme called C3 convertase. And C3 convertase, they'll actually work through these specific mechanisms. We're not going to talk about it. We're going to kind of keep this a little general. It splits C3. And when it splits C3, it splits it into 2 parts, right? So imagine here's C3. And when it breaks it into 2 different parts. One is called C3A, and the other one is called C3B. The part that breaks off and moves away is called C3A. The part that stays is C3b, okay? Then what happens? C3b will then allow for the binding of what's called C5b. Then C5b will be bound by another one called C6. Then it just keeps going down all the way to 9. C7, C8, and then C9. So again, what do we have here? Antibody mediated, binds onto this antigen, activates the Fc portion of the antibody, allows for this quick cascade of proteins, which is going to go in order. C1, C4, C2, C3b, C5b, C6, C7, C8, C9. Now, if you saw here, I mentioned B again. What do you think is going to be popping off over here when the C5 convertase works? It's going to be C5a. And I'll talk about what's so significant about these proteins in a little bit. Okay? Actually, you know what? I'll mention it now. All right, remember over here, we had the mast cells. They're going to be over here in this area, right? And remember, the mast cells are releasing a ton of different chemicals, right? Because of that inflammation. They're releasing histamines, they're releasing leukotrienes, they're releasing prostaglandins. Bradykinins are also going to be formed within that area too, right? It can also release molecules which are called proteases. And what these proteases do is they act on C3a and C5a and activate them, turn them on, right? So now look, C3a and C5a are active, right? And if these guys are active, guess what they do? They are great chemotactic agents. Now, if you remember, what is a chemotactic agent? Remember, someone throws a baseball at me, right? And if they hit me with the baseball, what am I going to do? I'm going to look that way. I'm like, who the frig did that? And I'm going to go that way, right? That's what these C3a and C5a molecules are doing. There could be a white blood cell right here, right? Could be a monocyte, could be a neutrophil, could be a macrophage, right? Or a monocyte, I'm sorry. And then these guys could follow that C3 and C5a. So what is C3 and C5a doing then? They're enhancing the inflammatory response. So they're enhancing inflammation. So they're enhancing inflammatory response by working through chemotaxis, right? They're helping the chemotactic mechanisms. That's that thing. Now, let's get back to this. C1, C4, C2, C3b. It starts this cascade, but when it does this, what can happen is C5b, C6, C7, C8, C9 can break off. It can literally break off. And if it does, look what happens. So imagine I take all of these 5 proteins and I make it into a protein channel. Look at this. So now look, I have the inner part of the channel. And then I break this up into segments here, right? Look what's here. C5b makes that part of the channel. C6 makes that part of the channel. C7 makes that part of the channel. C8 makes that part of the channel. And C9 makes that part of the channel. And if you imagine that, it makes this channel a pentameric protein that can actually break off. So all this part right here, I'm going to draw it in red actually, around all this part here, can break off and form this membrane pore that can actually bind into the bacterial cell membrane. What is this protein right here called? This is called a membrane attack complex, or also abbreviated MAC. What is the purpose of this MAC? Look at this. They're now fluids from inside the cell and outside the cell. There's an open movement between there now, right? So what's actually rich out in this area? Water. And if water is really, really rich within this area, maybe there's a lot of sodium out here, maybe there's a lot of water, these molecules can flow right in. They can flow right in into this actual bacterial cell. And guess what's going to happen? This bacterial cell is going to undergo lysis. This guy's going to get all fricked up, okay? So now look, the water's flowing in, the sodium ions are flowing in, all of this fluid's flowing in, and it's causing the cell to get bigger. And as he starts getting bigger, he'll undergo lysis. That's a beautiful thing, right? That's a beautiful way to kill the bacteria. All right, so now we've done the lysis mechanism. What else is actually interesting here? You see this exposed part here, the C3b? We can actually break this off. Now the C3b is exposed, right? So let's go ahead and erase this part here, and let's see what happens now. If that whole thing broke off, and now all we have left here is just C3b, because already we broke off C5b, C6, C7, C8, C9, right? We broke all of these things off here, and they're going to go and form that MAC. Now, this is what's awesome. C3b is a beautiful, what's called opsonin. Here, let me write that up here. What is that called? It's called a opsonin. Opsonin. What is an opsonin? An opsonin is basically, think about it like this. this bacteria, right? Think about it as just like French toast, right? But then I can take that French toast and I can put something over it that makes it even more tasty. Like what? Like Nutella, right? So I can take the Nutella and rub over that French toast with the Nutella. But now I just made it a lot more tasty and a lot more desirable to eat, right? Because nobody just wants to eat plain French toast. They want something's going to taste good. That's what these antibodies and all these things right here are doing to it. It's making the bacteria more tasty or easier to eat for who? For the macrophages and the neutrophils. Specifically though, the macrophages. So now look here. How does it do it though? So again, what was right here? This was also C5a, right? So again, look right here. We're going to have a macrophage right here. So here's this macrophage. What's on this macrophage that allows for him to be able to bind with that? A receptor that's perfectly complementary to that. That receptor is called it's an easy one, C3b receptor. And the C3b receptor on the macrophage will bind with the C3b here for this complement cascade, which is bound to the antibody, which is bound to the bacteria. And then what it'll do? It'll do the same mechanism that we had up here, that phagocytosis, where it takes those pseudopods and pulls the entire bacteria, as well as these complement proteins and this antibody, into the actual macrophage. And then what? The macrophage can do the same process it did before, where it breaks all of those things down and exposes it with the MHC2 molecule and the foreign antigen, right? The neutrophil can also do the same thing, but it will just exocytose those antigens. Ready? What happened again? C3b receptor on the macrophage, bound with the C3b for these complement proteins, which is bound to the antibody, which is bound to the bacteria, and it pulls it in and enhances the phagocytosis process. When that opsonin does that, the opsonin, which means making it tasty, can cause opsonization. And that will, what is the whole purpose of this? Enhance phagocytosis. So it enhances phagocytosis. So then you eat more bacteria, right? And if you eat more bacteria, you're going to be able to either secrete a lot of, exocytose a lot of those bacterial antigens or express them on the MHC2 molecules, right? All right, now, what is the overall result then? Again, what is the overall result of this right here? First thing is membrane attack complex. Second thing is opsonization. And actually there is a third thing, and that is the release of C3a and C5a, because that enhances the inflammatory response, right? This pathway that we just talked about that's antibody mediated and starts at C1 is called the classical pathway. Okay, this right here that we just went over is called the classical pathway. There's 3 of them, so we got 2 more to go. Okay, let's go into the next one. Let's come over here. So again, let's say that that's bacteria, right? Here's that bacteria again, right? Let's actually move this out of the way. And again, what's on that bacteria? This nasty bacteria. Let's say here's this nasty bacteria again, right? And it has, on its cell surface, it has those bacterial antigens, right? And again, look at this guy. He's holding his hand. Look what he has right here. He has a pitchfork, right? Look at this guy. He's mean, all right? So now, this bacteria, it's going to have specific antigens, which, you see all these proteins that we talked about, all these C1, C4, C2, C3, all that? There is that C3. He's so special that he can literally bind directly to the actual antigens. He can literally bind to the pathogen surface, straight to the antigen. So he can literally bind straight to the antigen. Who is this again? This is C3b, right? Because what will happen? You'll release C3a. And what will C3a do? It'll enhance the inflammation, right? So it'll enhance inflammatory response. So increase inflammation. Then, who will bind after that? It always goes C3b, then it goes C5b, C6, C7, C8, C9, right? So now, look at this, C3b, and then who also will be released from this? C5a, right? And again, that will become activated by the proteases, and then they'll enhance the inflammatory response. But if you remember, the same thing's going to happen. C5b, C6, C7, C8, C9, they can all break off. And if they all break off, they form again, what is that molecule right here called? That molecule is called a membrane attack complex, which can get embedded into this bacteria and can cause a lot of extracellular fluid to leak into this actual bacteria and initiate lysis. All right, and what else? If all of these molecules break off, right? So if these molecules, that C5b, C6, C7, if all of these guys, C5b, C6, C7, C8, and C9, if they all break off to go and form that MAC, that membrane attack complex, look who's exposed again. C3b. And if C3b is exposed, who can come back over here and bind? The macrophages. Or the neutrophils, right? Because what do those guys have on their actual cell membrane? They have C3b receptors, which are perfectly complementary, and these C3b receptors will enhance the phagocytosis process through a process which is called opsonization. Okay, so it's called opsonization. So what is this pathway called? Where you start with C3b directly binding to the antigen surface, and then you go C5b all the way on. This is called the alternative pathway. Okay? So now, the alternative pathway again, antigen, C3b, directly binds to the pathogen surface. There's no antibody-mediated effect here. Then, now technically there is another protein. I'm not going to go into super detail. I will mention him, but it's not super important. Technically, in between C3b and C5b, there is what's called factor B. So technically there is another molecule here called factor B, or BB protein, right? And factor B can actually, factor B will break off, but he can actually go off factor B, so it'll be C3b, factor B, and then C5b technically. All right, anyway, what's the overall effect? Their alternative pathway, they can initiate lysis through the membrane attack complex, or they can act through opsonization, which enhances phagocytosis, and at the same time they can release C3a and C5a, which will be acted on by proteases to enhance inflammation. That's the alternative pathway. Let's go into the last and final pathway here. Okay, so let's say here's a bacteria. And that bacteria, it's still that mean bacteria, right? So look at this guy. And again, what does he have on his surface? He has specific types of antigens. Right? Now this guy, look at him, look at his legs. He's so weird, right? All right, this guy right here, this bacteria. He has specific types of antigens on its surface. These antigens, remember I told you that antigens could be sugar molecules, they could be proteins, or they could be glycoproteins. This is actually a specific sugar molecule called mannose. It's a specific carbohydrate called mannose. Now, there is proteins that are actually within our body. One of those proteins is called mannose binding lectin. It's called mannose binding lectin. So what will happen here? There'll be a protein that will actually come out here, and he'll just so happen to perfectly bind with this mannose, and this protein is called mannose binding lectin. So there'll be this pathway right here. You're already going to kind of know what's happening here. This is the lectin pathway because it's not antibody mediated, and there's this molecule, this protein molecule called lectin that will bind and form this mannose binding lectin. It's actually mannose binding lectin. It'll bind there, and then we're going to initiate what's called the lectin pathway. So what is this called? The lectin pathway. This is our third and final pathway for the complement system. So now, after that guy binds, then what happens? Another different type of complement protein loves to bind to that mannose binding lectin complex. That is called C4. So it doesn't go C1, it goes to C4. And then who? C2, C3b, and then C5b, C6, C7, C8, and C9. And by now, you should already know what's going to happen. what's going to happen? C5b, C6, C7, C8, C9 will all break off, and they will then form what's called the membrane attack complex, which is a pentameric protein, which gets pushed into the cells, right, into this bacterial cell, into its cell wall or cell membrane, right? And then when that happens, what starts happening? A lot of these actual water molecules and extracellular fluid starts flowing in, and again, initiates lysis. So that cell will then be destroyed. What's the other mechanism? We already told you this, and you're going to know this by now. You will know the complement system by the time we're done with this, right, guys? So, again, what are these proteins here that causes the MAC? C5b, C6, C7, C8, and C9, right? All of these guys are going to form the MAC. But then if you remember, C3b is a great opsonin. So it's going to activate specific types of white blood cells, specifically like macrophages or neutrophils or dendritic cells. And what's going to happen? What do they have on their cell membrane surface? They have a complementary protein specific to that C3b. And this is called C3b receptor. And then what happens? It'll actually bind and cause phagocytosis, right? And then it'll actually do what? If it's a macrophage, it'll present it on its surface as an MHC2 molecule with that foreign antigen. If it's a neutrophil, it'll exocytose it into the extracellular fluid. So easy, right? Now, one more thing to talk about with this. Oh, and then what else? C3b, it also can release C3a, and C5b can also release C5a. These two molecules can be acted on by proteases released by the histamines, and they can enhance the inflammatory response, right? By chemotaxis. What did we just do again? What is the purpose of the complement proteins? Complement proteins are actually a part of our innate immune system. In other words, it's non-specific. Okay, we're still not even into adaptive yet. So these are proteins produced by our liver that are normally circulating within our plasma, made inactive, but then are activated during this inflammatory response by specific types of changes within the actual hemodynamics or the microcirculation via the increased vascular permeability, and it can be acted on, or can work through either antibody-mediated mechanism, right, through the FC portion, and go all the way from C1 all the way to C9. If it's C5b to C9, it works through what's called a membrane attack complex, which is a pentameric protein, which can create a pore within the actual bacterial cell and cause lysis. If it goes to C3b, this acts as a good opsonin, and it can actually cause opsonization, where it enhances phagocytosis via the phagocyte C3b receptor. C3a and C5a can be acted on by proteases secreted by the mast cells, which can then convert C3a and C5a into the active form, which can enhance the inflammatory response via chemotaxis, right? And then again, this is the classical pathway because it's antibody mediated. This is the alternative pathway because C3b can bind directly onto the antigen or the pathogen surface. And then this last one is the lectin pathway because there's a mannose sugar which is bound by mannose binding lectin, and this initiates C4 and then C2, C3b, same effect, opsonization, membrane attack complex. That is the whole function of the complement system. All right, in our next video, I want you guys to click up in the right corner here. We're going to continue our inflammatory response, looking more specifically at another part of the innate immune system, that being interferons and toll-like receptors.